In this video, we're going to look at proofreading of tRNA and how tRNA interacts with its codons. Now, the loading of the tRNA with its amino acid is very accurate. For example, if you compare isoleucine to valine and adding the correct amino acid to tRNA isoleucine, you only get it wrong once every 40,000 transfers, which is really amazing when you look at the two amino acids, and the only difference is a extra CH2 group in isoleucine. If you do free energy calculations solely based on theory in mathematics, you would predict val is inserted for every 100 isoleucines. So this is telling us that there is some proofreading that is going on to make sure we put on the correct amino acid, similar to DNA polymerase 1 once we actually looked at the mechanism. So if we are studying or looking at isoleucine RS, we see that it actually has two active sites. Site 1 will activate isoleucine in valine, while the second site will only admit amino acyl groups smaller than isoleucine. So site 1, you can actually add in valine to the tRNA. Site 1 doesn't have any idea that, well, has some idea, I suppose, because it should be every 1 out of 100. So 99% of the time, you're going to add isoleucine. 1% of the time, you're going to add valine. And site 1 will do both of those. However, site 2 will only allow the tRNA with the valine attached to it to be entered there. And any amino acid that goes into site 2 with the tRNA gets cut. So what happens is that you remove the valine and the AMP in there as well. And what's happening is that the 3' end of our tRNA is shuttling between these two sites via a conformational chain. So we're moving to these two different sites to make sure we don't actually accidentally insert our valine. So we are adding our isoleucine with high fidelity, but we are using ATP. So that's our trade-off there, which is an okay trade-off because we don't want to have errors in our proteins. If we look at other amino acid tRNA synthetases, we can see other different ways to make sure the correct tRNA is being added, and these are usually done through non-covalent interactions. Now we're going to look at how tRNAs interact with codons, and proper tRNA selection is only determined through the codon and anticodon interaction. An mRNA codon and the anticodon will pair in an anti-parallel fashion. So codon's 5' to 3, then the anticodon is 3' to 5. And many tRNAs will bind to 2 or 3 different codons that specify their amino acid. For example, our tRNA phenylalanine has a GMAA. GM means we have a G but has a 2' methyl group on it. And this can recognize UUC and UUU. So this is actually giving us a little bit of flexibility in what codon we recognize. And this non-Watson-Crick base pairing can occur at the third codon-anticodon position. And when you look back at the anticodon or at the genetic code, we'll find that this third position is where our degeneracy actually happens. So it really doesn't matter for a lot of our amino acids what that third nucleotide is, they'll specify the same amino acid. And the way that this anti-Watson-Crick base pair happens is through the wobble hypothesis, which was actually proposed by Watson and Crick. Is that, and this wobble hypothesis is that the first two codon-anticodon pairs have normal Watson-Crick geometry, while the third pair has some wiggle room, some wobble room, if you will, where you can have limited conformations that allow for these non-Watson-Crick base pairs. And that's what's being shown here, where you have different bases that can interact with different anticodons, because there's just more flexibility in the geometry of that pocket to allow for these non-Watson-Crick pairs. And looking at codons a little bit further, if we study yeast used in baking, we find that out of the 61 codon triplets, really only 25 are commonly used. So some codons are used more than others. And the preferred codons are those that form our Watson-Crick base pairs to our anticodons. So they're mostly or nearly complementary to our anticodons and the most abundant species in each set of tRNA. So these codons, not surprisingly, since they're used more, that means the tRNAs are more abundant as well, and these tRNAs have these perfect Watson-Crick base pairs. Same thing happens in E. coli, but the codons that are preferred are different from those in baking yeast. And what this means is that our genes can have different levels of expression based on the degree with which they have these preferred codons. That is, a gene that has a higher percentage of these preferred codons is transcribed more. And this is important when you want to use organisms to grow protein. So if you're having a hard time growing a protein or getting a low yield, it might be that you're using rare codons. And so if you change your plasmid or your sequence to use these more prominent codons, you could get a higher yield of protein. Or something else you could do is you could just have the organism create more of these rare tRNAs for you, and then you should have higher expression as well. And that is it for this video, and if you're watching in 2020, that is the last video I have for you. So I will see you in the final exam. If you're not watching in 2020, there's probably more videos moving forward. So if so, I will see you in those. If not, I will see you in the final exam.